Hello, everyone, and welcome. My name is Eleonora Tegaray. I'm Ictus Marketing Director, and on behalf of the team, I would like to thank you for joining us today for the webinar AI Orchestration Drug Discovery from de novo design to automated synthesis. Before we start, the usual housekeeping. Throughout the presentation, we will be sharing relevant links and content in the chat. Please make sure to review. To ask questions, please use the Q&A feature, and you can do so anonymously if you want. The session is recorded, and you will receive the link via email within 24 hours. Please feel free to share the link with your colleagues. So let's introduce the speakers of the day. We will start by Frank. Frank Leveillon, he's a senior scientist and deputy lab head at Ictus. He has a master's degree from the École Nationale Supérieure de Chimie de Montpellier. And Frank carried out his PhD studies at the EPFL in Switzerland under the supervision of Professor Wasser. During his studies, he explored photoredox catalyzed functionalizations of radicals using hypervalent iodine reagents. He then moved as SNF postdoctoral fellow to the group of Dr. Cornella at the Max Planck Institute in Mannheim an der Ruhr, where he developed nickel-catalyzed CO bond formations using nitrous oxide. Frank returned to France to start the LabCom HeatCat, a joint venture between the company Sequence and the group of Professor Masson. Since July 22, he has joined Ictus as a senior scientist, deputy lab head, where he works on the development, installation, and utilization of the Ictus robotics platform for AI-driven drug discovery. Thank you, Frank, for joining us today.
Hi, everyone.
The webinar will be opened by Quentin Perron, Ictus Chief Scientific Officer and co-founder. Quentin is a medicinal chemist by training, and he holds a PhD in organometallic chemistry from the University of Geneva. During his postdoctoral fellowship at UCLA, he worked on the total synthesis of bradycardia A, a complex natural molecule known for having potent immunosuppressive activity. After working as a medicinal chemist in CNS indications at Laboratoire Servier, Quentin switched to data science and chemoinformatics at Kinton, a company specialized in data science services. In 2016, together with his business partners Jean-Gaston Maté and Nicolas Dohu, he co-founded Ictus. As CSO, Quentin brings his vision for innovative AI technologies to accelerate drug discovery, with solutions ranging from de novo drug design to AI orchestrated robotic synthesis, which we will be discussing today. Quentin, thank you for joining us, and the floor is yours.
Thank you, Noah. Hi, everyone. I'm very happy to be here today with Frank to show you our vision and how at Ictus we have developed technology to go basically from a list of molecules to make, coming from AI into a list of instruction and to pilot robot to, at the end of the day, obtain our molecules. So we have quite a packed agenda. So let's start with the 4 challenges of robotic chemical synthesis. As you know, discovering new drugs, going from a target to a preclinical candidate takes a lot of times. And it costs a lot of money with a low success rate. And there are 2 main questions you ask yourself in this process is what molecule should I make and how to make it as fast as possible. Right now, to answer those 2 questions, usually it's the role of the chemist, the ComChem, and currently molecules are designed mainly by medicinal chemists and computational chemists and made by chemists. So we ask ourselves this question a few years ago, is it possible to incorporate somehow robotic solution into that process, which is known to be traditionally handcrafted? And of course, being able to incorporate such technology in such a complex process, there are many challenges, and we'll go through those 4 main challenges we have been able to identify. The first one is what type of molecule are we talking about? Are we talking about very simple molecules, very complex singleton or complex libraries starting from advanced intermediate or somewhere in the middle? This is a very important question because if you want to build such a robotic platform, you want to know exactly where you want to be in that process. The second question is the diversity and the liberty you let to the system to run chemistry. Either you fix most of the part of the molecules and you allow very few type of chemistry and you are more in the field of library design, or you allow the system to use any type of exit vectors and function to react in a given position. And in that case, you're more in the field of drug design, rational drug design. The third main difficulties is when you have a big to-do list of molecules, how do you organize yourself? How do you organize your workload? How to make decision rapidly for those molecules? Which retrosynthesis route should I select? What starting material? How to contact the providers rapidly, easily to get quotation? How can I fill my ELN properly, rapidly? What happens if I don't receive some building blocks at some point in this retrosynthesis pathway? And how can I run the certain type of chemistry in parallel when, et cetera, et cetera. So you rapidly understand that it becomes very complex. The fourth main difficulties is the type of robot. Are we talking about flow, chemistry, batch mode, how to transform a protocol to robot instruction? Who does that? AI, scientists, chemists, how to track vial position to avoid mistakes, to make sure that the robot is doing what we expect in the good position, how to handle different chemistry on the same platform the same day with solvent, reactant, reagent, etc. So again, these are important challenges. So what we have been able to observe is that, overwhelmed by all those challenges, the scientists usually used to make it simpler, meaning that they will design a very simple library of molecules with a limited chemistry, with one reaction at a time, encoded workflow. And usually this is what we can observe, is that you have a main scaffold, you have one exit vectors, for instance, here's the aniline, and you want to perform an amide coupling disposition. In that case, you will have a list of acid moieties, and it's very easy to automate that. In each vials, you have your starting material, your building block, the core scaffold, and in each individual vials, you will add a different acid moiety, and so that you can get a library at the end of the day. The problem of that is that this approach has a very limited impact because this is not what happened in the real project of drug discovery. And usually this type of approach, there is no adoption. And unfortunately, this is what we have been able to observe with the Lilly platform, L2S2 project, which was a fantastic project where they invest a lot of money in building this platform, which is now dismantled because no adoption by the chemists, no adoption by the classic workflow of drug design. So we asked ourselves the question a few years ago, is there new ways where we can use robot today, thanks to AI, thanks to different type of technology, which are now available. And what we wanted to do is to have a list of very different molecules with different exit vector, different chemistry. As a chemist, I don't want to limit myself because of the fact that I have a robot. I design plenty of molecules and I just want to make them automatically. So I want to have a broad molecular diversity, expanded chemistry. I want to access to more complex structures. So as you understand, I want to go from a library design to a rational drug design. And if you want to be able to do that, you need to break different type of silo. The first one is you need to make sure that the molecules you're designing are feasible on robots. So you're designing molecules which already takes into account the robotic constraint. Then once you have a good molecules, you need to make sure that you are able to find all the routes which are compatible with your robot. Then you need to select the best one among all those routes, and then you need to execute that. And for that, you need to break the silo to make sure that all those independent technology can communicate together. And this is what we have designed at Ictos, where we provide into the end of chemist AI solution to be able to integrate those main pillars. Namely, we have Machia to design molecules which are feasible, Spire, which is able to identify routes, Ilaka, which is organizing the workload and transforming a list of smiles, a list of molecules into a list of instruction. And then we have our robots, which can run that into the platform. So Machia, quick explanation of Machia. So first, Machia, it's not just a screening, it's GenAI. Contrary to what you can see out there, Machia does not generate smiles. Machia generates molecules taken into account building blocks and reaction, because we have trained Machia not on a database of smiles, but on a database of reaction. Machia is trained on 6 million reactions, so Machia knows organic chemistry. So when we design molecules, we design novel and feasible molecule by design, and we can explore huge chemical space, today estimated to 10 to the power of 27 possible molecules. Of course, we are not generating molecule randomly. We can generate molecules following a certain type of fitness function, like 2D QSAR, presence, absence of substructures, descriptors, but also 3D score like docking or bolts or other type of scores. Micah is a software as a service, which has a very simple interface. The goal is to be used by chemists and CompChem easily, and we can already incorporate robot constraint into the generative AI process. The second technology called important to mention that we already done a webinar a few weeks ago or months ago on Micah. So feel free to scan the QR code here and to get access to all the content and also the recording of this webinar, the papers we mentioned, the one we published. You know, it's a very it's a lot of information, so you can take time to go through it. The second important technology we have developed is Spire. Like Micah, it's run on organic chemistry, 6 million reaction. So contrary to Micah, which takes starting material and reaction to design a molecule, Spire is doing the reverse. So you input the molecules and Spire will tells you how to make it across different type of chemistry and building blocks. Of course, Spire can be customized to make sure that you find routes which are compatible with your robots. Spire is a data-driven approach, so it can be easily trained on a new data set of reaction, a new set of starting material, so you can totally customize Spire given your ELN, your constraints, your robots, whatever. And it's publicly available on Spire.ai, so you can test the Spire. In less than a week, we can retrain Spire, retrain the different AI models which are embedded into the solution, and at the end of the day, you have a GUI, you input the molecules, you can explore the trees. Spire is also is one of the application which has a lot of advanced search options. So when you run Spire, you can constrain the system to a certain price of building blocks, to delivery time provided by different type of providers. You can also play with a number of steps in your retrosynthesis tree. You can define also the name reaction and the chemistry you want to use during that retrosynthesis search. And this is one of the cornerstone of our robotic platform, because you can force Spire to tell you if this molecule is feasible using robot or not, because you can constrain the name reaction during that search. You can also define the key intermediates, the presence, absence of substructures, and you can also define the bonds you want to break during that retrosynthesis, or the bond you want to see untouched, like the bond you want to protect. Spire is available through GUI, as I explained, but also through API, so that you can connect Spire to external system. You send smiles, molecules to Spire API, and Spire API will perform a retrosynthesis, and it can send you back either the route, either a score, either a number of steps, or any information you would like to see. And this is a type of technology we have embedded in our ILACA technology that, Frank, we're going to present to you, how we transform a list of molecules we want to make into the robot into a list of instructions, passing by compound ordering, etc., etc. And I will let the floor to Frank.
Yes. So the third solution that we have developed is called ILACA. And ILACA has two main roles. It's allow us to develop new reaction type and new chemistry on the robot, and also to allow us to run a campaign of reaction. By this we mean synthetic campaign, where we want to isolate the final compounds. So in ILACA, we have actually integrated proprietary scheduling AI technology, and this allows us to organize a workload. We also have a procurement system that is making us in contact with external provider, and we can automatically drive the robot by structuring the necessary data in a machine readable structures. Overall, with ILACA, we are actually minimizing the human intervention in the lab, and we try to actually deliver this through a user-friendly interface for chemists in order to have a better adoptions of this kind of technology. So one of the first questions that arose when we thought about ILACA is how to transfer the procedures that we get from Spire, so from our retrosynthesis software, and how we can transform this in information that will be readable by the robots. And actually, through natural language processing, it was feasible to get most of the information in a very well-structured pattern. However, we wonder if this was actually enough. One of the first questions is it, can we trust the pattern protocol? Is it a reliable procedure? Also, can we trust the AI extraction? So maybe it's due to sometimes the way we can write palladium catalyst in a different patent or the different reagent. You can have some mistakes that will happen, and it's difficult to be 100% that the extraction will work perfectly. And finally, and most importantly, it's is this extracted procedure will be compatible with the robot constraints that you have in your laboratory? And this is a very tricky case. And so proposing reaction condition is something that is currently a hot topic. And for example, Coley and coworkers, they try to discuss this in the recent paper here. And in our case, we decided to not go through machine learning and data extraction, but actually to go through expert-based templates. And like this, we have developed actually our own chemistry compatible our own chemistry that is compatible with the robot. For that, we need to remove all the different complex setup. And like this, I mean cryogenic temperature, very reactive starting material, but also it could be just some material that we don't think it's feasible to be suitable for a good dispense. So for example, hygroscopic starting material like fluorine, potassium fluoride. And finally, with PIA, it's possible to actually exclude some reaction. And like this, we can exclude the reaction that will generate radioisomer, and that will actually fasten the purification after all in the process. Importantly, we did all this work in an expert-based fashion and thanks to literature search. But we also, in this way, were able to introduce batching and clustering parameters. And by this, we mean that we were able to say that the Suzuki coupling may be compatible at between 60 and 80 degrees with a certain catalyst and a certain solvent. And we also have a duration parameter that is, for example, here 4 and 8 hours. And by doing several versions like this, we will be able to be very flexible on a Suzuki coupling, but also we will be able to couple a different Suzuki coupling in the same reactor with, for example, another type of name reaction. It could be a bug valve hard disk elimination. If we do this for all the different name reactions, we end up with actually a series of templates that are expert-based that we validate on the platform. So we know that the reagents that we will use will be well-suited and well-dispensed, that they are safe. So it's actually making our life much easier after all. So far, we have developed more than 65 name reactions, and it's something that we are always working on. So ILACA is able actually to help us developing this new type of chemistry. It's basically transforming a name reaction in suitable robotic setup. So we are able to assign the name reaction that we want to define for this template. We can define the reaction and reagents that we want to use, and also the solvent and the key properties like the number of equivalent, the final concentration, and very importantly, the key robot action. So at the end of the process, it's possible, for example, to make a filtration through SPE process. And this is something that we can just activate or not activate directly in ILACA. Everything is saved, and this will help us to automatically fill this reaction condition in the reaction review later on in ILACA. As mentioned, we are having so far 65 name reactions and correspond approximately to 130 templates. This allows us to have a better flexibility when we're making reaction review and to be able to actually validate reaction on the robot. This is continuous work, so every month we try to add more and more name reaction to actually enhance this toolbox. Just to give you an idea of what type of chemistry we're able to masterize in the laboratory. So most of the palladium coupling are feasible according to our setups. So from Suzuki to block valve Hardwig to Sonogashira coupling. We also perform a lot of copper catalysis, but as well as ester formation, sulfonamide bond formation. It's a very classical SN2 or SNAR reaction, also very often undertaken in our laboratory. And recently we introduced some oxidation and reduction reactivity. This is really particularly helpful, and also protection and deprotection, because in a case is very common. Finally, during some work in the pipeline of drug discovery, we had to introduce some heterocyclic chemistry, and actually a lot of things are feasible here. We already successfully attempted three of them, and that was able to unlock some chemistry on our robot for the pipeline. Very importantly here is how this workload will be organized by ILACA. So once you submit a list of molecules through the list of smiles, it could come from a design that has been made through Micah with a synthesizability constraint, or it could come from any design, a MedChem team, for example. When you have this list, you submit it in ILACA, and it will call SPIA API in the backend. In this case, you will get actually a return that you will not see, and there will be a lot of synthetic route that will be found by SPIA. SPIA then can make a selection. So we apply a second layer that is called scaling AI, where actually we will try to compare all the different routes. We will score them. And based on this quality score, we can try to get the best one and to manage at the end to have only one route per molecule. The idea here is to ensure that one of these routes for the first molecule will be possibly compatible to be run in the same reactor with, for example, the route number 1 of molecule 2. And if we do this like this will actually get a possibility to work at higher capacity in our robot. So overall, what we are doing is transforming the list of molecules here in one route per molecule based on the quality score and on the feasibility to intersection of these name reactions. At the end, because it's quite challenging even for AI, we are submitting this to the chemistry review and validation. And this actually will help us to validate or not what has been proposed. So we are trying to consider this as a recommendation problem. In the end, what the chemist will do is to submit this list of smiles in SPIA, define the retrosynthesis constraints, so the number of steps of retrosynthesis, the price of the building blocks that he would like to use. But it could be also like if there is a Carol center or other detail like this. Once he's ready for retrosynthesis, he will call SPIA API, and we will get the list of smiles. The chemist will then submit all this reaction to reaction review, and he can select a template. So in this template, we have automatic filling of all the information that has been embedded inside. And what is left to do is actually to adapt and adjust the scale of the reaction and the stoichiometry of the different components. If the chemist is happy with this, he can approve this directly, or he can reject, and we will reloop these molecules. After passing the reaction review, the compounds will advance a little bit in ILACA, and actually we will be able to make a quote of all the chemicals that are pending. So this will actually send directly an automatic email to the supplier, and the supplier will need to actually give back a price and a delivery time. We have the same process for compound ordering. In this case, as the supplier will need to fill the barcode of the pre-weighted vials. After that, we will receive these compounds in our laboratory, and it's basically a rack that we can submit directly inside of the Hamilton. This Hamilton is actually a smart fridge where we can scan all the different vials at once. So this is making actually the process of reception very convenient. Automatically, we have a connection via API, so between the Hamilton hardware and our ILACA software. And this will update our inventory instantaneously. When we are ready for a campaign, we can also use a smart fridge to retrieve the vial that we need for the specific campaign. The fourth solution actually that we've developed is a Ictos Robotics chemistry lab. And in this case, we are using automated platform for making synthesis up to 96 reactions in parallel. We can masterize more than 65 organic reactions, a different name reaction inside of our platform. And in less than a year, we were able to actually set up more than 3,000 reactions. We apply it in several drug discovery projects, and we were able to contribute with 300 final molecules, average synthesis in 2 steps. The success rate is basically around 60–65% over these 2 reaction steps. You need to consider that we are not making purification in the intermediate, so we are making a sequential 2-step reactions. And overall, we try to manage a very convenient workflow with a user-friendly interface so that one chemist can operate one robot. This is how our ChemSpeed platform looks like. So you can see the two reactors here and the substrate vials here. Basically, this robot can make two very important steps: the inverterization. So there is a gripper that can hold and move vials. We have the barcode scanner here that is able to actually read all the scanner and then allow database operation. We can also perform all the classic steps of organic chemistry. Basically, it's dispensing solids and liquid, heating up or cooling down, stirring and shaking, and also filtration or even evaporation. When we receive the ChemSpeed hardware, what was going along is actually the AutoSuite software. This AutoSuite software is a very nice software where you can tune and design everything that you would like to do. However, it's very expert-based. For example, for just moving one powder of this rack inside of the reactor, this is 16 parameters. If you go from a solvent dispensing between this position here to the reactor, this is even worse, it's 40 parameters. So this, if you want to use it on a daily basis, is quite challenging. And we wanted to try to simplify it and actually fix some parameters once for all. So our solution was actually to encode all these different parameters in macros, so with programming tools. And we were able, through using database operation, but also creating a different dynamic zone, to find a way to actually transform a complex sequence that need to be handled every day by an expert by only one program that can actually encompass all the different campaign synthesis we would like to do on the robot. So to do so in the macro, what's needed to be done is actually to create a fully parameterized functions where you have some database operation and hardware operation all together embedded in once. In this kind of macro, you can either iterate on the number of reactions that will use these functions, or when you are at the end of this number, you can directly go to the next macro. So by the succession of macro, you can actually make a program where you will be able to make all the different chemistry that you want to do on the robot in only one program. And you need to also have CSV files with all the data that will be necessary to be fed in this program. So where we run a synthesis campaign in ILACA is shown here. We try to make it a good tool for actually being able to handle multiple projects on the same platform. So imagine you have 3 drug discovery projects running together. You have 96 molecules that are ready to make. You have all the being work in your lab. However, you don't want to start the chemistry for only this project, but you would like to make most of it of your platform. You have different exit vector and 6 named reactions. In ILACA, we will just submit all this in the tactical schedule, and ILACA will give us this as a result. So ILACA will actually find everything that is suitable in reactor 1 and everything that is suitable for reactor 2. It will set 2 different time and temperature, so you are able actually to make the 10 bromo-Suzuki, the bromo-Buchwald, and the SN2 alkylation at 80 degrees for 10 hours. And in the reactor 2, you can make the other 3 named reaction at room temperature for 16 hours. So these 2 reactors are independent in time and temperature, and then it's possible to batch inside of these reactors different named reaction. This is a new synthesis paradigm that we are trying to work on and to demonstrate. And later, Quentin will show you how we got some good results on that. When we're ready to launch campaign of reactions, we have all the information that will be gathered together, and they will be able to be pushed in the database that will be sent to the robot. We also make an action planning tool that is very convenient to give a chemist a way to follow what's happening in the robot. So is this really helpful for translating from virtual world to the reality when you're loading the robot? So you have all the different compounds, the barcode, the quantity, and you have tool to prepare stock solution and filtration. Overall, our process involves the loading of the robot at first, and then going back to ILACA, pushing the data to start the synthesis. All this data will be sent to the Ictos Cloud database that is in connection to our robot computer, and this will be able to send execution steps. During the synthesis, we are able to collect information from the robot, and this will be pushed back to ILACA. Everything is then saved at the end of the process in our automatic CLN. Finally, the way we are working is actually to submit all the list of molecules in ILACA. Thanks to a SPIA API, we get back a list of reactions, and this list of reactions can be either accepted or rejected by the chemist. If we reject it, we can send it to a CRO or classical chemistry, or we can also do it manually in the lab. Otherwise, all the reactions that we'll go through will be continue in our orchestrator, where we can go for procurement system and reaction scheduling. When you are ready for performing the reaction, ILACA is able to structure the data and send information to the storage and also to the chemistry automated platform. At the end of the process, we were able very soon to connect this actually to the analysis and purification platform that we are trying to build very soon. This also will push the data that is linked to the campaign in the ELN, and we can save all this information. Importantly, what we have seen is actually during the different phase of a project, of a medchem project, is that during the hit discovery phase, 80% of the molecule are able to go directly to the robot and 20% to the CRO. During the hit to lead phase, this number is reducing to 40%. And in the end, in the lead optimization, we are arriving at about 10% most of the time due to complexity of the reactions. For that, for the two examples, I will hand it over to Quentin to show us the different results.
Thank you, Frank. Yeah, so we wanted to perform some kind of a stress test of the platform, trying to run parallel synthesis of totally different molecules. Here, 135 molecules. The goal was to see how the platform can handle that. We use Machia to generate those molecules using our in-house inventory, our in-house chemistry. And the goal was to generate a library to maximize the diversity of the structures. So on the next slide, if you show that if you take those 135 molecules and you give it to ILACA, you can see that you can make those 135 diverse molecules using 27 different type of name reaction using 231 building blocks. As you can see, very different chemistry. You give that to ILACA, and automatically, as Frank already shown, that the system can tell you that to make those 135 molecules, you need 3 campaigns of 11, 29, and 95 reaction. And each campaign is using 2 reactors, so 2 isotones between 10 to 100 degrees. And overall, this campaign, we obtain a 70% success rate. We obtain a purity between 90 to 100%, and we are preparing a nice paper, which is going to be published at the end of September. So keep posted. We use also this platform not only to generate a library and to show how diverse the chemistry can be run on the platform, but also to try to discover new starting points on new project. And here we were interested in the PKMYT1 project. It's a kinase with a lot of competitors working on those targets, especially Repair Therapeutic, in silico medicine, Evaris. They are all exploring the same chemical space with the same scaffold, and we say maybe this could be a very nice example to showcase the power of this platform. In less than 3 months, using our generative AI Machia, SPIA, the robot, we have been able to identify 4 novel potent hit series going from 30 nanomolar activity to 800 nanomolar activity. So a very nice proof of concept showing how fast we can come with new chemical matter with very, very high activity profile already at early stage. So not only we are using this platform to perform some proof of concept, but also for in-house pipeline with our flagship project called MTHFD2. It's to target autoimmune disease. It's a very complex target, and where we are very close to PCC, and finger crossed, maybe at the end of this year we should obtain our first preclinical trial articles. And now I will let the floor to Frank to show you in action how ILACA is working.
Okay, so this is our software, Ilaka. It's a single sign-on software, so you have your private environment, but also it's a very collaborative environment. So you have the name reaction here. In this name reaction, we are making a distinction between the lab name reaction and pistachio name reaction. So it's why we have some analogs, because the pistachio are often more specific. Then to the lab name reaction, we are actually making a link with a template that I mentioned earlier. So we are actually giving the information of some reaction condition that will be used on the platform. So all this is actually the engine of Ilaka. You can see here that there is a stoichiometry and some function group information here. You have the information on the different reagents, for example, here sodium carbonate, the number of equivalent, and where it's located. So here it's a powder. You also have the information on the solvent and the final concentration of the reactions. You can also have actually the different actions that are registered. You can activate it or enable it. And here it's dilution, SP filtration, or additional wrenching. And very importantly, you can also make a quenching on only one well if you want. Very important is actually this batching parameters, so the time range and the temperature range. This will be helpful to make the clustering later on in Ilaka. So the way we are actually making the retrosynthesis is through this strategic schedule. So either you submit directly from Makia, or you can get your list of smiles. The compounds can be very different, like here, or it could be actually a library design. We are able to adapt to any workflow. If you have common intermediate, it's possible also to submit them. So you can assign the project on your smiles, validate them to make sure that everything is correct. If you have the name reaction that you prefer to work with, you can select them. Otherwise, you can select everything. And now we're also able to make presets. So just a list of different name reactions. For example, the robotics lab name reaction that has been validated on the robots. You can import also a smile of the common intermediate if you're working on the library design. And you can also play with different parameters, such as the max steps of your retrosynthesis, the early stopping score, so the quality of your scoring retrosynthesis score. And you can also play with the price of your building blocks. When you're happy with this, you can submit this to a static request, and we are calling in the backend SPI API. In this case, we are able to run the high throughput retrosynthesis of all these compounds all together at once. And for these 9 compounds, it can take only a few minutes. And we already applied it to 200 reactions, and it was taking more or less an hour. When you have the final result, everything is displayed in Ilaka. It's possible to get the disconnection and the equation. You can have the reaction smiles and the pistachio name reactions. It's possible to navigate very quickly inside, so you can download a report with the success rate of your retrosynthesis. And if there is some compound that has not been found, you can make quickly a final selection there. And this helps us to reloop all these compounds. Then we are ready to make the reaction review. Here again, you have the different reaction that you would like to validate or not. You can see the reject button. And so if you directly see a bad disconnection, you can reject it. And if you think that it's worth checking it, you can review these reactions. This process takes usually 1 minute. What you need to do is verifying that the name reaction is correct and assign a reaction template. You can see the different list here that we have available, for example, in aldehyde reactive amination, and directly submit the information in the kind of ELN-like tab. Everything is automatically filled, and the chemists need to actually choose the limiting reagent and adapt the scale. When you are playing with the scale, you can see that there is this green bar, and this is actually directly linked to the volume inside of your robot. So we are using 8 milliliters vial in our apparatus, ChemSpeed apparatus. And if you go higher than 4 milliliters, this is quite forbidden for safety reason. And we actually need to make an operation here to decrease this value. It's possible to use, to adapt, obviously, the stoichiometry, so the number of equivalent of your different compounds. And at the end, if you're happy with what you have seen, you can approve it. We have an expert mode where we actually can modify any of this information, but we try to avoid this. The idea is really to expedite the workflow from a list of molecules to a list of instructions for the robot. Here, we are actually calling either the procurement system or internal check to verify if this compound is already available in our inventory. If you accept the procurement system, then we will push it to our new way to procure this molecule. We are working with CaseSpace as provider, and we have a tool here that actually is making all the information in a quotation tab. And we can submit this as only one quote. So we can create either here when we were making a selection, or we can automatically create a quote with all the remaining compounds inside. So you decide which provider you want to use, and then you can select the different compounds you want to quote. Automatically, we will send an email to our provider. So here in this case, it's going to be ChemSpace. And if we are playing now the role of the supplier, this is what he will receive. So he will receive an email with a link that has been sent by our Ictos procurement system, and it will be able to open this interface. This has been nicely designed by our team, and this is actually able to gather all the information that are necessary to make a nice quote, and at the end to make the purchase order. So you can select or unselect the different line. The provider can register the price and the delivery time. You can also apply shipping fees. Then, when the quote is submitted, all this information will be gathered in Ilaka. This is a very fast process. And now we can see that it has been quoted. As soon as this is quoted, we can review this quote. So if it's like this in a green light, it means that actually the price was normal compared to our expectations that we are mentioning at the very beginning, and we can directly accept. When you accept all these quotes, then you are able to make a purchase order of the compound that has been quoted. Again, here you need to indicate a maximum delivery time. So if you don't want to have your compounds delivered in more than 8 weeks, you can mention it to the provider. This again will send an email automatically to the provider that he will receive and open and go back to this interface. That now is a little bit a change, because in this case, the provider will need to fill the barcodes on the barcoded vials. The supplier will actually fill these compounds with indicated mass. So we get, in the end, pre-weighted barcoded vials. 1D on the side and 2D on the bottom. And he will also update delivery time. Once ready, the order is submitted, and they will ship the compounds to the lab. Obviously, if there was a different smiles, he can inform us, and we have a tool to automatically calculate and change the value that he needs to be incorporated inside. So if, for example, you're working with amine, you can have, for example, the hydrochloride salts that could be available, and this is taken into account in Ilaka. As you can see, Ilaka is already updated, and we know that these building blocks are incoming, so they have been shipped by the provider, and we already gather the information on the barcode and the quantity. And we are making the reception in the lab. Either you use it here, like manually, but we can also use Hamilton Smart Fridge to make directly this reception. As soon as we are scanning these compounds, either manually or via the Hamilton, we can directly update Ilaka. So this is taking less than a minute in each time. You may sometimes just need to refresh your window. As soon as these compounds are actually now in stock, it means that it will unlock other things in Ilaka. So we will show here that the reactions are moving from pending status to a schedulable status due to the reception of the building blocks. When the building blocks, when the reactions are actually now schedulable, it's possible to make what we call the daily schedule, the tactical schedule. We have 3 ways to do it. Either you make it through a manual selection. So here are, for example, the 6 reactions that are available. And you can see that we are able to automatically fill virtually the robot on the right. It will gather all the information that are necessary to be run on the robot in this way. So this is one way of making the batching, but we have two other ways. The second one is a supervised reaction schedule. So for example, if you have a lot of work to organize and you have potentially 2 labs or 2 robots, you can assign it to a different lab or different robots. So the process we're trying to put in place is actually very scalable. It's possible also to make filtration in this case through different projects, but it's also possible to make a filtration via name reaction or via time or temperature range. This is just to help the chemist being really the master of his work and not just dependent also for all the time on the AI solution. So in this case, it could be very convenient when you want to set up particular sensitive compounds or for classified reason between different projects. What is important here is also that we have this automatic daily schedule, and we are actually gathering all the schedulable reaction and trying to put them in the minimal amount of reactors. In this case, there was 6 schedulable reactions in 2 clusters. We applied it already on this 135 reaction, and it's how we ended up with a first schedule at 95 reactions and 2 other clusters. In this case, you can see that the cluster number 1 has 5 reactions, and there was 3 different types of name reaction inside. On the cluster number 2, there was 1 reaction. In this case, you can approve or reject your reaction schedule. So it's also upon the decision of the chemist to keep working on this schedule. So it's really a recommendation problem that we're trying to build and just to assist the decision of the chemist. But in the end, we want to have the chemist having the hand on it. So as mentioned here, we have the 3 main campaigns that we have developed in the POC. And the biggest one was 95 reactions. So the robot was completely filled. We can push the instruction for the campaign when everything is ready. So through the database, we have instruction for simulation to make it a first trial virtually on the robot. And we also have this action planning that I mentioned earlier, where we have all the information on the substrate, on the powder, how it's going to be filled inside of the robot. So then the chemist is not lost when there is a large campaign that is arriving. In this case, we also developed this tool to prepare the solution where you just need to actually fill a few information, and that will help the chemist to prepare this stock solution very rapidly. So in our process, in our robot, we have 47 positions of stock solutions. So yeah, we can sometimes in very large campaign prepare very conveniently 30 reaction in a very quick matter of time. Everything can be exported. It can be printed. So all we have here is saving the information back. So everything is saved in ILACA. Once everything is ready, we can load this campaign. At the end, we can complete the campaign and submit back the information file from the robot. And everything will be gathered back in ILACA, so we can move on to the next step. And then we will have the information on the intermediates, on the final products. And basically, we can also export an analysis instruction file where we have already pre-calculated different ions for LCMS analysis. This is very convenient when you are working on this high number of reactions. We are also making a solubility report to try to gather information that are actually lacking in the world of automated synthesis. So I think this is more as the demonstration of ILACA software. And later on, we will be able to integrate LCMS analysis and report directly inside. So we will have all the information on this all the spectra inside of ILACA, and this will help us to manage all the chemistry inside in only one place. Okay, and I will share again my screen. and I will give it to Quentin.
Yeah, rapidly where we are now, as you understood, we have Micah for generative AI, Spire for the retrosynthesis, Ilaka for the orchestration and the robots, ChemSpeed robots, and purification analysis and testing right now is outsourced to external providers, and this is something we're going to incorporate soon. We have been granted from the EIC. There are more than 1,200 participants. They select only 70 projects, and we are among those ones. So we have a grant of 2.5 million to integrate this purification platform. And they're also willing to invest in our next fundraising at the level of 5 million as equity. So we are quite proud of this grant. And if you want to test the platform, it's very simple. You can share a list of molecules. We can run Ilaka to show you which molecules we can make on the platform, the one we cannot. And if you want to go further, we can also run the chemistry on our platform. And if you are convinced and you're interested to go deeper, and we can also integrate this technology to your system, to your robots, integrate it to your software, your ELNs, whatever. You can also, for instance, replicate, if you don't have robots yet, replicate the Ictus robotic platform on your premises. So this is something also we can provide. And finally, we can co-develop something new with you if needed. So thank you very much. We are more than happy to take questions, and I think Eleonora, the floor is yours to deal with the Q&A section.
Thank you, Quentin. Thank you, Frank, for the presentation. It was really interesting, and the Q&A is very lively. So what we will do is that I will be reading the questions in the order in which we received them, and we hope to answer in time. If not, we can go a few minutes over if you can stay with us. So the first question is around the Spire platform. What are the current limitations? Would it generate asymmetric roots proposals or just leverage chiral pool of starting materials? How often is your commercially available starting material database updated?
So in Spire, we use a chiral pool. So we are not performing asymmetric catalysis. And the database is updated on a regularly basis, something like every quarter. The providers share with us their new data sets. Spire is to identify the roots, but then in Ilaka, we rely on aggregator, where we place quotation and order, and they come with updated price. This is why we have this process of quotation in Ilaka that Frank presented to you, to have really the updated price at the moment you place the order.
Thank you, Quentin, for the answer. Very clear. So now we move next to a question from Mathieu Gentil. He says, very nice talk. Are you optimistic to one day being able to make tricky reactions compatible with your platform? Cryogenic, bully, sensitive catalysis, carbonylation? And if no, does it have an impact on the size of the explored chemical space?
Yes, thank you for the question. So for cryogenic temperature, for example, it's possible on some different platforms. Our platform is actually suitable for -20 degrees to 150 degrees, but you need some very specific cryostat to run these cryogenic temperatures reactions. So it's feasible, but in our hardware, it's not feasible. However, very importantly, we are able to actually design the routes with robot constraints. So we are able to actually search for alternative routes that will not go through this specific reaction that you're mentioning. So it's possible that it will not be an issue for the chemical space that we are exploring.
Yeah, and on top of that, you know, it can happen that for some intermediate, you need butyl lithium or whatever. And as Frank mentioned, Spire will try to find a pathway avoiding that. And this problematic will arrive for the providers, which will have to deal with this type of chemistry and then ship to us a key intermediate we can assemble in our platform. This is why it's very important to have this connection between GenAI, retrosynthesis passing by your chemistry to avoid this type of problem where you design the molecules and then when you see how to make it, you need to pass by something which is not compatible with your platform.
Thanks, Frank, on that. Now we move to a question from Sylvain. He asked, how would you take solubility of certain materials into account when applying your pre-developed robotics protocols?
Yeah, so in our workflow, we need to solubilize first this building block and transfer them into the reactors. So obviously, if we are able to have kind of a model of making a link between the structures and the solubility in one of the solvents, this will be very helpful. So far, this kind of data is really scarce. And this is why we are building this solubility report at the end of our campaign. And so far, we have been able to very often transfer most of the compounds inside. But we know that maybe we have a lower yield sometimes due to solubility reasons. But this has never been a strong, the main issue, let's say, in our process.
Thanks, Frank, for the answer. So now we have two questions that are around root design. A first one from Stefan. I think, Quentin, you touched on this, but maybe you can expand a bit more. How does this work with new compounds without a known synthetic route?
And I think this is the beauty of Spire, that Spire can find roots of molecules which has never been described before. So, and it's related as well to the next question of obtention of the synthetic root of design compound. We should import synthetic root of molecules that have similar structures, and you are not obliged to have a similarity structures. Again, you can input a totally new molecules, and Spire will try to find a way to make it starting from commercially available building blocks in a reaction which can be undone on the robotic platform.
Thank you very much. So now we are changing gears around productivity. So Mathieu asks, how many chemists are required to manage the platform and the entire workflow? And he asked, including purification. Just to have an idea about the FTE equivalent versus the 3,000 reactions you perform.
Yeah, so as you have seen at the final slide, so we are a team of only 3 persons in the lab. We have our lab head, Jean-Christophe, myself, and Clémentine. And usually we are 2 that run the robots. And we are not at the cruising speed, let's say, of our lab. So it's possible to have increased number on that. So it's also a matter of how much product you are dealing with. And this purification and analysis, it's externalized so far. So this part of the work is not on our charge. But very soon we will have this new platform, and we're also continuously trying to integrate more and more this hardware. So it will be an automated platform that will involve a new liquid handler and HPLC machines. So everything will be tried to be as automatic as possible. And in this case, we will hopefully have only one more person in the team to work on that.
I think those 3,000 reaction has to be taken with a grain of salt, as Frank explained, because we could have done more. But during that period, we are first using the platform to develop new chemistry. So not necessarily producing molecules, but just to use this robot and develop new stuff. So it's also for us like a sandbox. And also we make molecules for our own projects and own endeavor. We are a small company, so we are not using it to produce a lot of molecules every day. So for sure, we could have done more, but this is not the goal of the platform right now, to do plenty of molecules, but more to showcase how we can batch on the same platform totally different molecules from different projects and different chemistry.
Thank you very much, Quentin and Frank. So now Kara asks, well, she says, first, very nice talk. I would like to understand how you manage the building block logistics, delivery, format, timing, etc. And how do you do the purification of the products? And you've touched on this, but in the logistic piece.
Yeah, on the logistic, so everything is handled actually through Ilaka. So we have the information on the delivery time. We can make the quote and purchase order. And when we receive everything, so we have this smart fridge Hamilton apparatus. We can make the reception very conveniently through API that directly update our inventory system in Ilaka. When we want to retrieve the vials for making a campaign, we just need to gather the list. So when we create the campaign in Ilaka, we can create a list of vials that are required to be run in this campaign. And we can just start actually the Hamilton from Ilaka. And the Hamilton will retrieve all these vials and prepare them in a rack. And those rack, we can submit it in the robot. So everything is quite integrated for this building block management, I would say. And I think there was another question on the purification level, but we discussed it quickly already.
Yes, thanks, Frank. So now we move a question around actually doing projects. Are any customers using Ilaka and robots in-house, or are all Ilaka customers using Ictus services? Also, for in-house Ilaka use, do you provide different procurement options to integrate directly with customers' procurement and or their preferred providers? And lastly, are you entertaining custom training models and customer preferred reaction libraries to expand the reaction options?
Okay, so first, our customer Ilaka is for Ictus and our in-house project, but we also propose AIDD collaboration where a client is coming with the targets, and we use our GenAI, our structure-based scores and technology, and of course Ilaka and the robots to discover new starting materials, new starting points, and to run the project from start to the PCC. And then the question is, so we are not selling properly Ilaka as it is as a software for now, maybe in the futures, or at least in the framework of a collaboration and a deployment of the robotic solution. And are you
Yeah, the last point was around custom models trained on preferred reaction libraries to expand their reaction options.
So I'm not sure to understand the question. Yeah, so we are not using the. I'm not sure to understand the question, but so we are not using the data of our clients to train or retrain our system. So right now Spire is trained on Pistachio database, so provided by Nextmove software. So it's patented chemistry. So this is enough to perform retrosynthesis for most of the chemistry we need. And then the reaction we are developing our expert-based system. This is Frank, Clementine, and GC who are developing this chemistry on the robots, and then save that as a template, as Frank explained during the webinar.
Okay, many thanks for the reply. So now we are changing gears around the type of compounds that the platform can handle. So Berthony asked, what is the maximum molecular weight of the compounds that are designed and synthesized?
Typically, it's small molecules. So we are ranging from, I don't know, 200 to 500 or 600 most of the time. But we are actually considering making new works on PROTACs type of molecules. So we think it's possible to actually go a little bit higher than what we are doing right now.
Thanks, Frank. So then we have a question on IP. With your robotic synthesis, are there any considerations or limitations regarding composition of matter IP?
I'm not sure to understand the question, but in terms of generative AI, when we generate molecules, we make sure, of course, that we'll be able to generate molecules with freedom to operate. So this is more control at the level of the generation rather than at the level of the chemistry.
Thanks, Santa. So the next question takes us to the very early stages of the process around the novel design. They ask, what is the workflow to optimize multiple properties, such as binding and selectivity, using your platform? And do you optimize one property at a time, or do you optimize all properties simultaneously?
So this is really the topic of the Makia webinar we done previously. So you should scan the QR code or just go to the link provided by Eleonora during the webinar where we explain everything. But basically, yes, We optimize molecules, again, plenty of properties from 2D to 3D, and we optimize everything simultaneously, including the synthetic access, of course.
Thank you. So then the next question is, can do you peptide chemistry using your robot?
And yes, it's feasible, but currently there is robots that are making peptide chemistry, I would say, more efficiently than our robot. Our robot is really made for making very different type of chemistry in the same reactors. So we are making amide bonds, but we are not trying to make very efficiently large tetramers or I don't know, hexamer. So yeah.
Thanks, Frank. So now we're going into space of purification, but it's a really interesting question. So it's a great talk. What is your approach and strategy towards high throughput purification? And are you planning on using SPI or MACA for predicting preparation methods on LC or SPE?
I don't know if you want to go on it, Quentin.
No, vas-y.
Yeah. We obviously it's interesting to try to apply AI models for having better separation. It will not be used directly in SPI or MACA, as this information is not embedded there. But as soon as we will build our platform, we will gather this information. As mentioned in ILACA, for example, everything is event-based, so we have all the details there, and we can directly focus on these informations. The approach here is actually to have a liquid handler that will be placed after the robot for chemistry. So then we are not taking the time of current robot. We need to have one robot for chemistry, one robot for sample preparation, and then the HPLC machine that will run the purification. And we also are working on the provisioning on a very nice tool for evaporation in parallel of all the fraction at the end of the purification.
And yes, we want to be able to predict the preparation method, not using MACA and Spire, but to use models to be able to select the good column, giving you know the molecules and the quality of the purification. This is important because the final target of this platform is at some point being able to develop an autonomous lab at some point. So each data which can be gathered will be gathered to build in the future good models.
Thank you very much. So now we have another question that also looks at the future applications. So have you tried this platform for process research to reduce the cost of active pharmaceutical ingredients?
We are working on it, developing ways to perform HT high throughput experiments, trying to balance, the good catalyst quantities, etc., to obtain the best yield. Because we need at some point that when we develop new reaction on the platform to have a reaction condition which are versatile enough to tackle different type of environment, I would say molecular environments, being able to perform Suzuki coupling in an electron-rich, electron-powered indoor environment. It's important to test different possibilities. And for that, we have decided to develop this type of option. So it will be, of course, used for API development.
Thanks, Quentin. We have a question that I'm not sure I fully understand, but maybe you do. How
Yes.
can we analyze all the reactions at one time? Like how much SM is left?
So currently we have not analysis in line. So obviously we are waiting at the end of the process to check what is the result of a reaction. It's possible to implement HT96 well plate inside of the robot and to try to submit this alipot. So we can actually prepare this plate and submit all at once, but it's going to be at the end of the process, or at least independently during the process.
Thanks, Frank. So now we have a question from Christopher Muller. He's asking around the business model. What is the business model to access the platform? Maybe you can share a bit more content.
Yeah, as explained during the webinar, there are different modalities. One is, trying to access if you want to develop such a robotic platform on your premises, we can replicate Ictos infrastructures and technology, to replicate that on your environments. We can also co-develop together something totally new, or integrate our AI and system into your environment. So we are quite open right now. And of course, if you want to start progressively, you can simply start by sharing a listing of molecules, and step by step, we can show you how we move forward with this listing in order for you to be convinced that this approach is new and efficient.
Thank you, Quentin. So we have a following question from Pascal George. He asked around the amount of samples that you can deliver, and he has a follow-up question around the analytic data that you're generating, LCMS, HNMR. And he has a third question on how do you eliminate organometallic impurities coming from catalysis at the final step.
Okay, yeah. So the amount of sample at the end is usually between 1 milligram and 50, depending on the success rate of our reactions. We have the trigger of 1 milligram because at the end, what we want to do is a bioassay sample, and we also put a trigger of 90% of purity for making this bioassay. For the second question, it's mainly LCMS analysis, and if required, we can also make more characterization with NMR, but this is costly for a small company like us. And finally, in the end, we can eliminate organometallic impurities via the filtration through SPE. And in this case, it's possible to use some metal scavenger in the SPE. And most of these catalysis traces or impurities are removed directly. So this is very convenient. And again, it's usually not a very big issue in our case when we are making this sequential coupling, because at the end, we are trying to get very robust condition to make this sequential synthesis.
Thank you very much, Frank. We have a follow-up question. Cara earlier was asking about building block logistics, and now she says, as an add-on, what happens when things are delivered weeks apart from each other?
And this is obviously a very good question. And this is the beauty of ILACA. So since we have our inventory that is updated every day, as soon as we have a reception, we are able actually to get the information of which reaction we can perform today. So as soon as all the building blocks or routes are there or available in the lab, the reaction is moving as a schedulable status. And this is how we are actually very easily managing the reaction we can perform today, or the ones that we need to wait a bit more.
That's why you have this tactical scheduling, daily scheduling. Every day you ask ILACA, given my to-do list and given what I have in the fridge, what can I do today? And this is totally automated.
Thank you very much. So we are up to our last two questions. Jan asks, is it possible to obtain the list of named reactions that were successful on the ChemSpeed platform?
I think Frank already shared it during the webinar, but we can elaborate a bit more on that, but feel free to contact us.
We have the contact details there. We can follow with you, Jan, also after the webinar. And our last question, it was mentioned that synthetic routes are about 2 steps. Is this correct? And what is the maximum number of steps that you can envisage?
So in average, it's a 2-step synthesis sequence. But actually, we managed, I think our maximum was 5 without making purification at each intermediate. So this is quite rare. And we try to prefer staying between 1 and 3. This is actually giving a better success rate at the end.
Thank you very much, Frank, Quentin. I think with this, we reach the end of the webinar. Thank you for all attendees that really stayed to the very end and the last question. You will be receiving the webinar recording link that you can share with colleagues. We have the contact details in our chat. Please feel free to reach out to the team if you would like to know more about how to adopt the platform.
Thank you, Noah. Thank you all.
Bye. Thank you. Bye.
Bye.